This station is called Spatter Patterns. Every criminal leaves behind evidence at the crime scene. In this project, scouts are going to be correlating the size of the stains to the distance they fell to solve a mystery. We're going to investigate how the size of spatter patterns change. We're going to investigate how the size of the spatter changes depending on the height that the blood was dropped from. In crime scenes where people are wounded, investigators apply principles of blood spatter analysis by taking measurements such as the diameter to figure out what happened. So to begin with, you're going to demonstrate what is blood first made of. So most people will say it's made of water and the red blood cells. But it's a little bit more than that because we have some additional things in there. So you have it in your container. You're going to go ahead and make some fake blood. First thing you're going to do is 2 tablespoons of cornstarch. And the cornstarch is sort of the proteins that are in blood, or represent the proteins that are in blood. So you have some white blood cells and you have some additional proteins. It's sort of the sticky stuff that's in there. And then we're going to add 2 tablespoons of water. And blood does have a fair amount of water in it. So we can go ahead and put that in, and stir that until it's nice and smooth. And talk about what else is in blood. We have our red blood cells, the platelets, to give you that nice red color to it. But it also has a thing in it called plasma. And the plasma is actually what makes blood a little bit sticky. So you can ask them, have you ever cut yourself and had some blood? And it's watery, but it also leaves a sticky. The platelets are what help your blood clot after a cut. If it was just water, it would just continue to flow and flow and flow. So to represent our platelets or our plasma, we are going to go ahead and add in some cornstarch. And we're going to put in 4 tablespoons of our liquid cornstarch. The doctors can actually take plasma out of blood by running it through a centrifuge, and plasma is very useful for people who have lost a lot of blood. They can actually do transfusions with plasma. It contains a lot of the proteins and platelets and other things needed within the blood. It also gives it a little bit of cohesiveness. So we have a nice blood solution. It's a little pink at the moment. We need to add a little red food coloring to it. You're going to add about a teaspoon. And then we want that blood to be nice and smooth and even colored. So add that in. We want to make sure it gets nice and dark. You can see I'm trying to make it so that there aren't any streaks of red, but rather it's all one consistent color. We'll go ahead and let that sit for a few minutes while the scouts are getting their papers ready, because in order to test the patterns we got to know where it's being dropped from. So each scout should be in teams of 2 to 3 scouts. And you're going to give them 4 pieces of cardstock. And then they'll also need a ruler, an eyedropper, and when you're ready for them, you're going to give them a little cup that has some fake blood in it. So first thing you want them to do is to label their cardstock. So we'll have some pencils or pens available. And they're going to label it number 1. Is 6 inches. So once they have it labeled, number 1, 6 inches. Number 2 is 12 inches. And this is the height by which they're going to drop their blood. Number 3 is 2 feet. And number 4 is 4 feet. We want to make sure that all the partners get a chance to drop some blood, so they can either split it up so that one scout does the 6 inches and the other scout does the 12 inches, or that they do half and half so that each scout gets a chance to do it. They're going to make 10 drops of blood. So they can go ahead and put it in their eyedropper. We're going to drop it from 6 inches. We'll move over, and they're going to make a total of 10 drops of blood on each of their papers. Notice I'm moving the ruler a little bit out of the way so that the blood doesn't spatter all over the ruler as well. And if they need to refill with their blood, they certainly can. And you can see at 6 inches, it just makes a little splat. Next, they'll go ahead and do the same thing at 12 inches. And again, we want it to be a nice 10 splats, because that gives us a nice scientific database. The hardest thing that's going to be for them is to just do one drop instead of releasing their entire eyedropper full. You can see from 12 inches our size increased just a little bit. You can compare it, the 6 to the 12. Our next one will be at 2 feet. This is a little large, but you'll get the idea. Your kits will contain a number of yardsticks and rulers for the kids to be able to do this. One of the things that the kids should notice at 2 feet is that it starts to spatter a little bit, so you'll get micro dots next to their large dots. So again, you can see our dot size has increased, and also you're starting to get some of the spatter where it's making the smaller dots around the larger dot. And then their final one will be at 4 feet. Because the fake blood does not dry particularly quickly, I would not let scouts take this one home unless they have just a great desire to. Again, if their adult is around, I would have them explain that really it needs to sit out to dry. It will eventually dry. It takes a long time though. And their final one is from 3 feet. And from the furthest away, it makes the largest of the dots. So now you can take a look at them. You can actually even have them measure their dots on here. So if I were to look at my 6 inches, for example, I can see that it's about 1/4 of an inch. Or if I look at my from 3 feet, my dot size is almost 1/2 an inch to 3/4 of an inch. So the higher the drop falls, the more it spread out as it hit the paper. You could help your scouts graph the results if you wanted to, if you had some extra time. So it looks like so. Six inches, 12 inches, 24 inches, and 36 inches. And then look at our size. We have 1/4 of an inch, 1/2 inch, 3/4 of an inch, or an inch. At 6 inches, the average size was about 1/4 of an inch. At 12 inches, our average size was a little bit larger than that, so between 1/4 and 1/2. Or if you wanted to be scientific, you could go along to the metric and actually do it in centimeters for graphing purposes. Get them used to looking at both sides of the ruler. At 2 feet, it's about 1/2 an inch. And then at 36 inches, we had 3/4 of an inch or a little bit higher. So you could show them a nice little bar graph on how the height correlates to the size of the drop. The next question that you could ask them is, what happens if you're not standing still? What would happen if they drop the blood at an angle or while walking or running? So in each of your kits, you'll have a larger piece of poster paper, that you can have a couple of kids come up and demonstrate what would happen if they were walking or running while they were dropping the blood. So you can have them walk down, and while they're walking or running, they can be losing blood. That's where you'd have them squirt the entire time while doing it. Or if you're walking slowly, it might look like that. What if you got hit at an angle? Makes a different kind of a drop again. So you'll be able to demonstrate then the difference between running, walking slowly, or getting hit off to the side. You can also test that with the ruler. They saw what happened at 12 inches being dropped this way. Makes a spot like that. But if your paper was standing up, like you were on a wall, and you were out 12 inches, which would be approximately here, if I gave it a good squirt, it makes a very different pattern. Let's see if they can come up with any other ideas as to what you could be doing where you might have a blood spatter. One thing with the pronunciation, it is not splatter. There's no L in there. It is called blood spatter, and it is part of forensic science. So it is something that is being used that scientists study currently. So you can bring in that real world connection and career connection.